Hello everyone. In this class, we'll learn about erythropoiesis, that is the production of RBCs from the bone marrow. Formation of red blood cells is called as erythropoiesis. In normal adult human, the site of production is bone marrow, that is red bone marrow. In the first 3 months of intrauterine life, blood cells develop from mesoderm of yolk sac or area vasculosa, that is in mesoblastic stage. After 3 months up to 5 months of the fetal life, red blood cells are developed from the liver and spleen, which is called as hepatic stage. From 5th month onwards up to the birth and up to the adulthood, it is from the red bone marrow and it is called as myeloid stage. In children, erythropoiesis occurs in all bones with red bone marrow mainly, and liver and spleen. And in adults, erythropoiesis occurs in red bone marrow, which includes the end of the long bones, that is, shaft is converted into yellow marrow, as we know, and it occurs in flat bones like skull, vertebra, ribs, sternum, and the pelvis. Different stages of erythropoiesis. Erythropoiesis, that is generation of the red blood cells, mainly occurs from the hemocytoblast. It is considered as a pluripotent stem cell and is non-committed, and the diameter of hemocytoblast is around 18 to 20 microns. It is nucleated with a thin rim of basophilic cytoplasm. The nucleus may contain two or more nucleoli with open chromatin. They proliferate extensively and give rise to committed stem cells. Committed stem cells, they develop from the pluripotent stem cells. There are two types of committed stem cells, myeloid and lymphoid series. They have become committed to give rise to a particular line of cells, either erythrocytes, platelets, monocytes, like that. Committed stem cells of myeloid series give rise to all types of blood cells except for lymphocytes. Then they give rise to progenitor cells, which are developed from committed stem cells. The progenitor cells are of two types, the burst forming unit of erythroid series and colony forming unit of erythroid series. The burst forming unit gives rise to colony forming unit of erythroid series cells. Then they undergo into Proerythroblast stage, where the first blast cell, immature cell, belonging to the red blood series, is Proerythroblast. They are formed from the colony forming unit of erythroid series stem cells. They have a large nucleus, distinct nucleoli, and open chromatin, and the cytoplasm is predominantly basophilic and shows active mitosis here. In the early normoblasts, the size further decreases. You won't see nucleoli. The nuclear chromatin gets condensed into threads. The cytoplasm is basophilic here and it shows active mitosis. That is, they still proliferate. Intermediate normoblast stage, the cell size further reduces, chromatin thread further condenses, and hemoglobin start appearing in this stage. Cytoplasm become polychromatic, that is, it stains both eosinophilic as well as basophilic, and in the later part, the mitosis stops. The late normoblast, further reduction in the cell size occurs, nucleus moves to the periphery, further condensation of the chromatin thread occurs, and in the later part of the nucleus becomes pycnotic, and there will be further increase in the concentration of hemoglobin, and that is why the cytoplasm is mostly becomes eosinophilic. Reticulocyte, the cell size reduces and almost the same as that of matured RBC, that is around 7 to 9 microns. There is no nucleus in reticulocyte, and when stained with the dyes like brilliant cresal blue, cytoplasm shows a small reticulum, that is due to the presence of RNA, and hence it is called as reticulocyte. Cytoplasm here more of eosinophilic, and then it gets matured to erythrocyte. So erythrocyte is fully eosinophilic, non-nucleated. It is biconcave and the diameter is around 7.2 microns. So these are the different stages of erythropoiesis. They all derive from the pluripotent stem cell, that is hemocytoblast. So then they commit it, and then they get converted into burst forming unit and colony forming units to give rise the erythroid progenitor cells, precursor cells, which is nothing but the pre-erythroblast. And pre-erythroblast gets converted into early normoblast, intermediated normoblast, late normoblast, where the active mitosis will happen till the stage of intermediated normoblast, and hemoglobin starts appearing in the stage of intermediate normoblast. And gradually the size of the cell is going to reduce till it becomes reticulocyte and erythrocyte. The factors affecting erythropoiesis. They can be general factors, hormonal factors, and maturation factors. General factors. Erythropoietins. Erythropoietins are glycoprotein in nature and also called as hemopoietin or erythrocyte stimulating factor. It is secreted by the interstitial cells in the peritubular capillaries of kidney and by hepatocytes in the liver. Nowadays, it is thought that it is produced from the juxtaglomerular cells of the kidney. In adults, 85% of the secretion comes from the kidney and 15% from the liver. It acts from the stage of committed stem cell, and it causes early differentiation of erythropoietin sensitive stem cells to proerythroblast and subsequently to mature RBCs. It prevents apoptosis of red cells. It increases synthesis of RNA, DNA, globin, ferritin, which increases the heme synthesis. It increases the release of reticulocytes from bone marrow. It, it, uh, it stimulates the secretion. It is stimulated by mainly the hypoxia, androgens, cobalt, catecholamines, and alkalosis. The estrogen decreases the production of erythropoietin, and this is the reason for the reduced red cell count in case of females. So this is how oxygen is going to affect the production of the RBC. So it is going to stimulate the cells of the peritubular capillary or interstitial cells in the renal cortex or the juxtaglomerular cells and the liver Kupffer cells, where 85% of the erythropoietin production occurs in the kidney and 15% in the liver. So thus reduced erythropoietin, it acts on the bone marrow, it increases the RBC production, and the oxygen supply is restored by this feedback mechanism. Hormones, testosterone, thyroxine, corticosteroids, growth hormone favors, and estrogen inhibits the erythropoiesis by inhibiting the synthesis of the globin chain synthesis. Some of the hemopoietic growth factors, which are interleukins and stem cell factors, generally these factors induce proliferation of the pluripotent stem cells. Some of the maturation factors like vitamin B12 and folic acid, they're called as extrinsic factors and are necessary for DNA synthesis. Erythroblasts need them before every mitosis, and deficiency leads to failure of maturation and reduction in cell division. Iron is necessary for hemoglobin synthesis for the formation of the heme. Castle intrinsic factor, it is produced by parietal cells of the stomach, and it is essential for absorption of vitamin B12. Okay, it is also called as intrinsic factor. Dietary factors, proteins help in globin formation. The ferrous, that is iron, manganese, copper, cobalt helps in the heme formation. Vitamin C and calcium increases iron absorption from the gut. Note that androgens has a stimulating effect on erythropoietin, and estrogens decreases hepatic synthesis of globulin. The They also depresses the erythropoietic response to hypoxia. So that is why in females RBC count is less as compared to males. This is in brief about the erythropoiesis. Thank you.